What are stem cells? Stem cells are undifferentiated cells, meaning they serve no specific function. This is in contrast with nerve, intestinal, and heart cells, which specialize in the needs of their organ or body system. Basically, they do not have a certain job. Stem cells serve as a sort of a template for the creation of other cells. They are unspecialized and have no particular function other than the creation of new cells. In this video, Robert and I will discuss the two kinds of stem cells, embryonic stem cells and somatic stem cells. These cells perform a vital role in the human body because they are necessary for repair and regeneration of damaged cells. They help replace dying cells in our body by differentiating into a variety of cell types. The term differentiation refers to the process by which a more specialized cell is formed from a stem cell, leading to a loss of some of the stem cell's developmental potential. This means as soon as a stem cell becomes a specialized cell, the stem cell cannot go back to its original state and must stay as whatever it differentiated into.
Okay, so properties of stem cells. The classical definition of a stem cell requires two characteristics. The first is self-renewal, the ability to go through numerous cycles of cell growth and cell division while keeping its undifferentiated form. Stem cells maintain their population through asymmetric cell division. When a stem cell divides, there is always a stem cell along with the unspecialized one. This can be done through stochastic differentiation too. In this case, the mother cell splits into two specialized cells, while another stem cell splits into two new stem cells. Potency. This term is the ability to differentiate into an array of specialized cells. For the sake of word simplicity, here are the types of potency for this video. Totipotent cells are stem cells that can differentiate into any cell type. These cells are the first generation of cells that can form a completely new organism and are created with the fusion of a sperm and egg cell. Pluripotent cells are descendants of the original totipotent cells. They can form any cell type. Multipotent. They can differentiate into a couple of cell types in a closely related family of cells. Oligopotent. They can differentiate into a few cell types. And lastly, unipotent. They can only make one cell type.
The difference between embryonic and somatic stem cells. While embryonic and somatic stem cells have similar functions and characteristics, they have their differences as well. Embryonic stem cells are considered pluripotent, given sufficient and necessary stimulation for the cell type. Their function in the embryo is to form a new human body, and this requires them to be malleable, meaning they must be capable of differentiating into any or most cells in the body. On the contrary, somatic stem cells are multipotent. Their only function in the body is to create more cells of a given kind. For example, cells in the gut lining, skin, and eyes. Somatic stem cells cannot form as many types of cells as embryonic ones can. Where stem cells are found. Embryonic stem cells are found only in the undifferentiated inner cell mass of a human embryo. Unlike somatic stem cells, they can only be harvested in embryos, hence their name, embryonic. Somatic stem cells can be found in three main areas of the human body: the bone marrow, adipose tissue, and blood. Why embryonic stem cells are important. Embryonic stem cells are so valued in medicine and biology because of their unique pluripotent nature. The highly regenerative nature and plasticity of these cells are incredibly important to the field of medicine, genetics, and bioengineering. Embryonic stem cells have the potential to specialize patient care through stem cell therapy, give insights on how genetic conditions form, as well as the breakthrough of lab-grown organs and eradication of diseases from defective body parts. The controversy. The use of embryonic stem cells in research is controversial due to the fact that they can only be sourced from embryos. This presents a moral and religious challenge for those on both sides of the pro-life versus pro-abortion debate. For many who believe that human life starts at conception, the mass harvesting of embryos for embryonic stem cell research is basically a holocaust against the unborn. The use of human embryos is also controversial for more pragmatic reasons. It's hard to obtain industrial amounts of embryonic stem cells from embryos alone. Clearly, a better, more efficient method to source stem cells must be found. Potential applications for stem cell research. Stem cell studies can help advance knowledge on how diseases and medical conditions form as researchers observe stem cells differentiate. For example, researchers could simulate defective stem cells to form a bone that develops osteoporosis later on. This technology can massively help research into the biology of the human body and diseases, because it gives researchers a clearer picture into the inner workings of our body. Furthermore, stem cells can help us ethically test drugs for safety and effectiveness. Through lab growth of stem cells into specific tissues, we can pinpoint how medications will affect the body without putting any animals or peoples in harm's way. This growth and maintenance of stem cells in vitro may even lead to breakthroughs in food science, such as lab-grown meat.
Regenerative medicine. Lastly, embryonic stem cell research can revolutionize medicine and biology. To start this off, to treat failing organs, modern doctors usually request for a donor organ. These requests can take weeks or even months to be fulfilled due to a limited supply, with many patients dying without ever receiving a donor organ. This limited supply of organs also creates a black market for healthy organs sourced through shady means. Even if the patient manages to receive an organ, due to the organ being made of foreign cells, it is attacked by the patient's immune system. The immune system attacks all foreign cells, even human cells. So patients typically have to take immunosuppressants to calm down their body, but this also leaves them at risk for infection by pathogens. Stem cell therapy can help solve this dilemma. By stimulating cells to become liver cells, for example, doctors can inject a number of these cells into a fibrosis liver and regenerate the damaged cells. Doctors can even take this a step further and manufacture whole organs in case of a total replacement. This technology has the potential to save lives. Stem cell therapy can be used to heal a number of diseases, including Alzheimer's, Parkinson's, lupus, osteoporosis, and myopia. Any disease caused by malfunctioning cells can potentially be cured through the use of stem cells. First and foremost, the main issue with the progress of stem cell research are the moral limitations brought about by the harvesting of embryonic stem cells. This topic heavily plays into the abortion debate, whether or not human life starts at conception, birth, or later on in life. This is due to the fact that researchers must destroy human embryos to get the embryonic stem cells in the inner germ mass. We'll quickly peruse through the views of various groups. First off, we have the United States and the EU. Despite being traditionally Christian countries, they have become more liberal and secular since World War II. They are both known for their progressive policies and both have strict limitations on stem cell research for moral, religious, and political reasons. Next, we have China. As the Chinese Communist Party espouses state atheism and authoritarianism, neither religion nor public opinion is a concern for them. As a result of this, China has the most permissive stem cell research in the world. Judaism. Jews and the State of Israel permit embryonic stem cell research if the embryo has not been implanted in the womb. They believe that the Talmud's laws on killing infants do not apply on embryos in vitro. Christianity. The Catholic Church, Pentecost, Baptist, and Methodist oppose embryonic stem cell research. They see it as needless destruction of human life and as a sin. Many other churches also take a stance on opposing abortion. Islam. Islamic scholars have issued fatwas, or interpretation of Islamic law according to scholars, promoting stem cell research as long as little harm is done to the subject. Although with these fatwas they did not make a distinction between embryonic and somatic stem cell research. Islam believes that fetuses have souls after 120 days of gestation. Counterarguments for these complaints state that a 5-day-old embryo is too young for personhood, or that embryos and fetuses before birth do not have human rights at all. But if stem cell research should advance any further, research on alternative sources of pluripotent stem cells would be a great help. One way to source pluripotent stem cells is to induce pluripotency in somatic stem cells. This is done through the 4 Yamanaka factors with an in vitro culture. The 4 Yamanaka factors are basically proteins that induce somatic stem cells to return to a pluripotent state, meaning that they can turn into a wider variety of cells and they can be used in the same manner as regular embryonic stem cells. And secondly, as stem cell therapy is still in its infancy, it is not perfect. A common issue with using these fast-growing, rapidly changing cells is that it can create tumors. Patients of experimental stem cell treatments have had tumors in their body composed of multiple cells and many different kinds of cells, including tumors made of hair cells, bone cells, and heart cells. More research into how to control stem cell growth and what kind of cells they turn into will be needed before stem cell treatment can be used en masse. And lastly, scientists in the field of embryonic stem cell research. Shinya Yamanaka and John Gurdon both won a Nobel Prize in August 2006 for their breakthrough in stem cell research. They are the scientists who discovered the 4 Yamanaka factors, which are the proteins that can turn somatic stem cells into an induced pluripotent state. This is very important because it provides us with an alternative source of pluripotent stem cells that doesn't require harvesting of embryos. Here are our citations.